Dr. Kurt Schalper obtuvo su título de médico de la Universidad de San Sebastián. Posteriormente continuó con su doctorado en la Universidad Católica de Chile. El grupo del Dr. Schalper evalúa el papel de los antígenos tumorales específicos y las vías de evacuación inmune utilizadas por neoplasias malignas de pulmón humano, incluyendo ligandos y receptores coinhibidores, estimuladores inmunes, células inmunosupresoras. El Dr. Schalper ha publicado 50 artículos, de los cuales 40 son resultado de investigación original. Actualmente dirige un laboratorio de investigación en inmunooncología en la Facultad de Medicina de Yale. Recibámoslo con una gran bienvenida.
So the title of my presentation now is talking about biomarkers for cancer immunotherapy. And in this talk in particular, I will focus not only on clinically relevant biomarkers, but also in trying to understand what are the determinants for sensitivity and resistance to immunotherapy. This is just a picture of our beautiful cancer center at Yale. And these are my disclosures. So during this presentation, there's going to be three components. In the first part of the talk, I will try to convince you that immunotherapy is actually very important. And we will discuss what are the determinants for sensitivity and resistance, what of those are clinically useful, and which of them are actually approved for use in patient care. I will also specifically talk about the relationship between qualitative biomarkers such as immunohistochemistry versus more quantitative biomarkers and how we can be more objective and more reproducible. In the second part of the talk, I will center the discussion around non-small cell lung cancer, and I will show data to connect what happens between the tumor site or the somatic mutations and how that interact with the immune system by recognizing those candidate neoantigens and how this can actually influence response to immune checkpoint blockers. Finally, I will close the presentation by showing the future directions, which include how we are mapping the immune system in the tumor microenvironment to essentially recognize not only lymphocytes, but also myeloid cells, how we are measuring cytokines using RNA transcripts, and then what are new technologies that we are now including to have enhanced tumor immune profiling. So the most important point today is that among many ways to reinvigorate the immune system to recognize and kill tumor cells, there are two major pathways that have been proven to be clinically useful. And both of them are actually now being used clinically, and they are part of the therapeutic armamentarium. The first one, it's indicated on the left side of the slide, and it's called the CTLA-4 pathway. And this essentially is an immune suppressive receptor present in a number of immune cells upon T-cell receptor activation. And blockade of this inhibitory signal induces a stimulation of the immune cells to recognize and kill tumor cells. And there are currently a number of different drugs. One of them is clinically approved for melanoma and a few other scenarios to clinically treat patients. The second pathway that it's relevant and it will the most discussed during this presentation is called the PD-1 pathway. And as shown here on the red square is another immune suppressive pathway in which an immune inhibitor receptor called PD-1, which is also co-regulatory after T-cell receptor stimulation, also mediates an immune suppressive signal. What happens here is that many tumors overexpress a ligand for that receptor called PD-L1. And by engaging these pathways, tumors can evade immunity. So blocking of this pathway using monoclonal antibodies to, for example, PD-1 or PD-L1, so the ligand or the receptor, with a number of different drugs can actually produce a prominent anti-tumor immune response. And I will go now deeper into these determinants. One of the important points of these new immunotherapies is that as opposed to what we have seen before with targeted therapies in which we can affect a very small group of patients that have particular kinase mutations, this is a very different story. And this slide summarizes what we know today about different tumor types here, and the ones indicated in green are the ones for which there is already an FDA-approved indication. Then we have the ones in blue, which are the ones for which there is activity, but there hasn't been an approval yet. And then we have the ones in red, in which there is essentially very limited or no activity. But as you can see from this slide, the majority of tumors so far tested, they have response, and this is very unusual from cancer. The other important point is that you can see that many therapies are already approved, and they span a number of different cancer biologies. We find all the way from melanomas, which are essentially mesenchymal tumors, to straight carcinomas like lung cancer and bladder, but also neuroendocrine tumors like Merkel cell carcinomas and now hepatocellular carcinomas. So the most important lesson here is that these therapies are very important. They span a range of tumors, they're widely active, and arguably they are the most impactful therapies yet created for cancer. One of the important points here is that most of the last decade we have been focused on studying the tumor cells, and we have done extensively used sequencing to try to understand what happens with mutations, and now there are a number of metrics from this immune response that are related with the tumor. Some of them are indicated in here, and we actually use now PD-L1 expression in tumor cells, but also the presence of genomic instability, the mutational burden, in which I refer more to, but then neoantigens and other things. And actually, we can now capture some of the tumor metrics in peripheral blood. So a number of things happen in the tumor. But the most important point here is that it's not only about the tumor here. Now we also have to capture measures from the lymphocytes recognizing and killing those tumors. And there are a number of things that have been shown to be relevant as determinants of response. For example, the amount of TILs in pre-treatment samples and post-treatment, and they can be recognized using different methods. We can measure clonal expansion and contraction, and then other cell types. But the message here is that it's not only about the tumor, and no matter how hard we sequence and we identify mutations, if we don't understand recognition, determinants, and engagement of the immune system, this is not going to work. So now we have really a separate paradigm, and we have to take care about two sides. If we collect the literature from the last probably 5 to 6 years of what are determinants that have been reported to be associated with response or resistance to immunotherapies, we have a number of factors that I actually divided into more immune or phenotype markers that are indicated here and more genomic markers. Usually these ones are related with the immune system and these ones with the tumor. And we can see that at the immune markers we find PD-L1 IHC, presence of tumor-infiltrating lymphocytes, but also RNA signatures related with those lymphocytes. There's a number of data emerging about the gut microbiome as a major determinant for response, but this is not clinically useful yet, and then myeloid-derived suppressor cells and other immune-related signatures. Then from the tumor side, there are a number of genomic markers like microsatellite instability, the number of mutations, usually called TMB or tumor mutational burden, but also specific mutation in discrete genes that can decouple immune recognition. For example, we know about interferon gamma receptor 1, beta-2 microglobulin, MDM2, and a few other discrete mutations that can essentially make the tumor invisible to the immune system. There are also specific alterations in PD-L1 locus by DNA fish in Hodgkin's lymphoma that are very, very characteristic, and they mediate huge responses. And then also there are some metrics that can be captured at the immune level from the T-cell receptor sequencing. But what have we learned from all of this data? I think there are a number of conclusions. One of them is that there is prominent signal in the tumor. Most of these factors have been directly measured in the tumor, but there are emerging circulating biomarkers. So many of these metrics are going to be able to be captured by a simple blood test, and there are some examples of that already happening. The second important point is that having this many factors, can we actually integrate them? Are they all redundant or they're independent? And I will go back to that point during the presentation. From the clinical standpoint, the most important point is that only 2 of these factors are today FDA approved for clinical use. And there's actually one in each category. We have PD-L1 IHC, which is approved for a number of indications, but it's largely used in lung cancer. And then we have microsatellite instability that has been referred to by previous presentations. This is the current clinical standard, okay? A number of things are probably going to be approved over the next years, but this is what we know today. What is the relevance of biomarker? A lot of people say, you know what, this is very general. Why are we using biomarkers? And there are examples that biomarkers can actually be very relevant. This is a summary from 2 studies that were done independently using PD-1 axis blockers in frontline lung cancer. So that means that these patients with advanced lung cancer see first immunotherapy even before chemotherapy. Immunotherapy is the first therapy they get to treat their disease. And they actually used two trials. One is Keynote 24 using pembrolizumab, an anti-PD-1, and then Checkmate 26 using nivolumab, another anti-PD-1. For all purpose, those two drugs are believed to be equivalent. One of the important points is that, as you see in the left chart, pembrolizumab here was significantly better than standard chemotherapy, and the difference was actually very, very prominent. However, nivolumab was not better than immunotherapy in frontline lung cancer. One of the important factors here is that this trial selected patients based on high expression of PD-L1, more than 50% of tumor cells. In this study, only more than 5%. So although this may not be the only factor determining this difference, it may be one of the most prominent, indicating that using biomarkers can actually be useful for deciding what patients are going to benefit from immunotherapy. One important problem here is that, as opposed to the target therapy world in which we knew a specific mutation in a specific gene in the tumor cells, here the landscape is much more complicated. And this cartoon that was put together by Nicolas Restif a number of years ago now captures that complexity, and I actually look for a real human lung cancer that mimics this cartoon, in which we see a vascular structure here, a number of immune cells, tumor cells, macrophages, how all this interacts, how we make sense of all of this. And some targets for immunotherapy can be expressed in tumor cells, some are in macrophages, some are in vascular cells. So really, no matter how hard we sequence DNA, we won't be able to capture this complexity. So I think we need to go beyond and now try to make sense of the tumor immune microenvironment. The second important complexity here is that there are a number of treatments already being studied. And this is a summary including only for lung cancer, all the clinical trial combinations that are ongoing today, with a backbone of anti-PD-1 like nivolumab and pembrolizumab with all these combinations, and then atezolizumab and durvalumab, which are anti-PD-L1 with all these combinations. If you can see this, how are we going to interpret this data? We're combining immunotherapy with a number of immune and non-immunotherapy, and there are a number of targets like LAG3, TIM3, KER, CTLA4. They can be in different tumor cells or immune cells. So really, how are we going to make sense of this data? This is another important point. And there are today more than 1,000 different combination immunotherapy being tested clinically. So really understanding the tumor microenvironment may help us driving those clinical trials and make sense. The third important point is that to date, the clinically useful immunotherapy biomarkers are based on immunohistochemistry. And this is a representative picture from a well-differentiated lung adenocarcinoma, and we can see the carcinoma cells here forming gland-like structures. And this is a PD-L1 immunohistochemistry straight out from the clinic. And you can see here that this is a pretty easy case. So we see most of the tumor cells are brown, meaning that they have PD-L1 expression, but there is also a signal in the alveolar macrophages, a little bit in the stroma, and so on. But this is a very easy case. Most of the tumor cells are positive for PD-L1, so we could say this is 80%, 90%, or more than 50%. However, there are other cases that are way more challenging, and this is another example of another lung carcinoma in which we see scattered PD-L1 signal, and we are supposed to score only tumor cells only in the membrane. And when we start looking, we see that some cells have a little bit of kind of membranous staining, but this is really membranous or perinuclear. And then some cells look like tumor cells, but it actually could be a macrophage. And then the question is that 1% of tumor cells, 2%, 4% or 6%? And would pathologists agree on what is the percentage? And the answer is not. So the pathology is actually quite subjective, and the way we're scoring this today in the clinic is just having a pathologist like myself looking at a bright field image and estimating about this much. So this is not reproducible, and it's not quantitative, and that makes a lot of problems. So what we've been doing in my lab for some years now is to try to make this more objective and to do quantitative measurements. And one important point here is that being an emissive method, so fluorescence is much better at quantifying signal because we see light production from the target and not like absorbance, like immunohistochemistry. And this is an example of some we published a number of years ago now in which we validated assays to quantitatively measure PD-L1. This is our human placenta, which is a positive control, transfected cells, and then non-transfected cells. And we actually developed assays to measure PD-L1 protein or PD-L1 RNA in human paraffin tissue, and then we can collect the fluorescence signal and quantify it very accurately. And this is an example of the output of these assays in which we can now quantify, make statistics, and then we can reproduce scores over time. So we can actually have a now quantitative output. We have proven that these metrics are actually useful, and this is in the same study, we measured over 700 lung cancers. We colocalized PD-L1 with cytokeratin to highlight tumor cells, and we could actually see that some cases had a lot of PD-L1, some had very little, and the same with RNA. But then we look at outcome. And what we could see is that both PD-L1 protein or RNA measured quantitatively in the tissue were actually associated with better survival. And this is an example in two different populations with PD-L1 protein shown here in blue, and then we have PD-L1 RNA. So not only we proved that we could reliably measure PD-L1, but we shown that it's clinically relevant or at least biologically meaningful. After that, we got also very interested in trying to make sense of the immune microenvironment. So it was not enough to just measure PD-L1. So we expanded the study to move from a typical estimation of TILs using hematoxylin eosin to a quantitative multiplexing assay to measure different T-cell markers. And this is published 2 years ago now, and we did a multiplexing panel to measure cytokeratin in yellow for tumor cells, CD3 for T cells, CD8 for cytotoxic T cells, and CD20 for B cells. And as you're seeing here, two tumors that were high TILs, we could actually now separate them, and this one had a lot of T cells, and this one had a lot of B cells, actually producing what we know now it's a tertiary lymphoid structure. So now we can not only quantify, but we can also measure the architecture and understand the difference between different inflamed tumors. And then using quantitative immunofluorescence, we can actually carefully measure the amount of each of these markers. In these charts, each column is a patient, and we measure the markers in tumor and stroma by colocalizing with cytokeratin. And we can very accurately quantify the level, and then we can show that it predicts survival, and it actually outperforms TILs by H&E. So we have proven that this method works, that it's more sensitive than what we have been doing yet, and it's clinically relevant. So now, how we go forward, how we can understand more. The other important layer here is that as opposed to flow cytometry or cytoph or other assays in which we grind the tissue to obtain the cell populations, here we have the spatial context. And this is very important because as you can see here now, by looking at the tumor cells that are positive for green cytokeratin, we can now define what is tumor and what is stroma. And we can now measure very carefully signal within compartments. And we have actually seen that some treatments can introduce more T cells into the tumor rather than increase the absolute number. So really having this spatial orientation, it's a prominent value. So now I will tell you what is going on, what is this conversation between the tumor immune genomics and the immune composition of lung cancer. And to approach this problem, we actually generated a study in which we collected 49 patients with advanced lung cancer with these characteristics, all of them receiving immunotherapy. So all of them receive an anti-PD-1 pathway, and a few of them also got CTLA-4. We collected the tissue, we reviewed that it was enough tissue, and we performed whole exome DNA sequencing, and then we also performed immunoprofiling to look at the number of T-cell metrics. The most important point is that now we do something called immunogenomics, which is a little bit different from just calling mutations. So what we did here with the whole exome sequencing, we called the synonymous or non-synonymous mutations. This is the traditional analysis comparing tumor and germline DNA. But then we also use algorithms to do HLA typing. So we use an algorithm called AFLATIS, in which we can, by looking at the sequencing, determine what is the HLA type of that patient. And once we know the HLA type, we can actually look at specific mutations and see how they are predicted to fit the groove of the HLA peptide of that patient. And that's what we call the candidate neoantigen. So we know what of those mutations can be seen by the immune system of that same patient. And then finally, we look at specific variants in K immune genes. So to understand if those help the tumor evading the immune response or not. So we collect all these metrics. And then in the same tissue, we look at an immunofluorescence panel that captured a number of different functions. So one of them was the cytokeratin for tumor cells, CD3 to determine the amount of T cells. But now we actually look at granzyme B, which is a effector molecule for T cells, and proliferation marker of T cells. We know that T cells, when they're in good shape, they proliferate a lot or they produce cytotoxic enzymes. So these are markers of functional performance of T cells in the tumor microenvironment. And then we look at the amount of T cells, the amount of activation of T cells by looking at granzyme over T cells, and then proliferation of T cells. And then we integrated all the data and look at the relationship with outcome after immunotherapy. So the first important point, and that was actually mentioned in the previous talk, is that we could see that there was a significant positive relationship between the number of mutations in those tumors and the number of packages that patients were smoking per year. The relationship was statistically significant, but it's not perfect, as you can see, and some of the very highly mutated tumors are actually not heavy smokers, but there was significant correlation as expected. The other important point is that we look at the relative amount of mutations in patients with major oncogenic drivers. So we stratified lung adenocarcinomas into the ones harboring EGFR mutations, KRAS mutations, or lacking both of those mutations. And these are the proportion of mutation-specific variants that we found, which is typical for lung cancer. But we could see that, as expected, because of their connection with smoking, patients with EGFR mutations have significantly lower mutations than KRAS mutant or EGFR KRAS wild type, essentially supporting that patients that are not smokers are more likely to have kinase mutations, and those patients have less mutational burden. But then we also look at the candidate neoantigens per each group. And as you're seeing here, not surprisingly, we saw that patients lacking major oncogenic drivers had significantly higher levels of both MHC class 1 neoantigens and MHC class 2 neoantigens. One important point here is that the way we call these candidate neoantigens using sequencing is in silico. So we don't really have experimental data of those neoantigens actually being presented and recognized by the immune cells. So what we did here to sort of filter the noise of that possible assay is that we, for some cases, in a background of HLA-A2, we synthesize peptide, recombinant peptides carrying either the wild type or the mutant variant. And by exposing those peptides to these cell lines called LCL174 that are lymphoblasoma cells that lack antigen presentation capacity, we can interrogate if the peptides can actually bind, be exposed at the surface, and eventually be recognized by T cells. So it's a functional assay in vitro to demonstrate that the candidate neoantigens are actually neoantigens. And what you're seeing here is that we use this by flow cytometry. We measured the HLA-A2 after 24 hours of incubating with the mutant peptides, and we could see that about 60% of those neoantigens that were called by sequencing were actually being able to be presented at the surface of the cell, essentially validating that the majority of neoantigens that we're finding at the sequencing are actually capable of being presented. However, we noted that these algorithms are much better for some HLA types than for other, and we're actually now trying to expand these efforts to be able to filter more the signal to noise in these assays. This is a very busy slide, but captures what are the major genomic findings in those patients treated with immunotherapy. And what you're seeing here in the upper panel are the number of mutations found in all these 49 patients treated with immune checkpoints. And then each patient that benefited from therapy or had durable clinical benefit, meaning response or stable disease lasting more than 6 months, is indicated with a red star. So as you can see right away, we see that most of the patients that show response to therapy are enriched to the left side, meaning that they were in the patients with high mutation, which was mentioned in the previous talks. So we see that patients with high mutational burden are actually more likely to benefit. But we also see patients with few mutations and actually in the very low end that respond to therapy. So this is not a mechanistic relationship. Some patients, even having low mutations, can still benefit. The other important point is that in the middle panel, we stratify the candidate neoantigens by strong, intermediate, and weak affinity, just by the calculated nanomolar affinity to the HLA type of the patient. And we can see that there is a strong correlation between the number of mutations and the number of neoantigens. The other important layer here is that we can recognize the phenotype of patients that are smokers, because smoking usually introduces C2A transversions, which are indicated with yellow. As expected for a population of lung cancer patients, the majority of cases had a predominance of C2A transversions. However, the two patients with the highest mutational burden did actually had a lot of C2T transversions, which supports the notion that the patients with the very, very high end of mutations may be a consequence of DNA repair problems rather than smoking. What happens first? Is the smoking hitting some DNA repair genes, or just the DNA repair genes had a mutation? It's unknown. But the most important point is that we look at specific mutations in DNA repair genes, and we could see that most of the patients with high mutational burden did actually have mutations in key DNA repair genes, like POL E, POL D, mismatch repair genes, and so on. So we're starting to see a connection between defects in DNA repair deficiency and then increased mutational burden and likelihood of response to immunotherapy. So again, what happens in the tumor has a reflection in the immune system, and this is what we're showing here. The other important point is that studying the relationship between these genomic metrics and response to therapy. And what you're seeing here is the number of non-synonymous mutations or candidate neoantigens in patients that show durable clinical benefit or non-durable benefit. And as shown by other groups too, we see that the patients with benefit or responders are actually significantly higher number of mutations and candidate neoantigens. And this actually translates into a better progression-free survival, but not so much in overall survival. And this was initially very counterintuitive. Why would patients that have better response and better progression-free survival not survive longer? We didn't know exactly, and we essentially were concerned that this was a small retrospective cohort. But a new study, actually at Checkmate 26, a prospective clinical trial showed comparable results, which opens the possibility that tumors with high mutational burden are actually more likely to adjust to immune pressure because they have genomic instability. So this is an open question, and we'll have to figure that out. What is actually, how the benefit translates into overall survival? The next important question is, were they actually mutations particularly associated with a higher likelihood of response and or resistance to therapy? So we actually stratified the patients by responders in the upper table and non-responders and look at those genes that were more frequently mutated in each group. And what you're seeing here is that there were a number of genes significantly associated, but only 2 of them did actually survive after full discovery rate adjustment. And those are TP53 and TRIM64B, which is actually a very, very unknown gene that we haven't been able to find a lot of biology behind that. The other important aspect here is that these analyses are very tricky because we know that the responders, by definition, are hypermutated. So the likelihood of having genes with mutations is important. So that's why we usually look at the replication time, because we know that genes that are very long or late replicators are more likely to be hit by mutations. But we also could see that a number of mutations in specific genes were associated with absence of response or resistance to therapy. And the top one was EGFR, which not surprisingly, because we know from the clinical trials that patients with EGFR mutations benefit less from immunotherapy. But we could also see P53 and then KRAS, but a number of other genes that we're now starting to look more carefully, particularly related with potassium and calcium channels. So some metabolic thing is going on here that we're trying to figure out next. The most important point is that we might be able here to identify pathways to mediate primary resistance to immunotherapy. The next question was to understand if there was any relationship between loss of heterozygosity across the whole genome or exome and response to therapy. So we actually plotted the LOH as shown here in responders and non-responders, and we could not see a significant difference, suggesting that LOH is not a major factor to response to immune checkpoint blockers. And then we also look at the number of both copy number gains and losses in relationship with durable clinical benefit or non-durable benefit. And although we could see a trend towards higher response rate in patients with copy number variations, it was not statistically significant. But I think further studies will need to be done to show if this is actually a metric that can be used or not. The other important point is that we knew that patients that express a lot of PD-L1 are more likely to benefit. So one important point was to rule out if those patients that we were picking up with genomics were the same that were expressing high levels of PD-L1. So to do that, we actually analyzed the cases with the 22C3 assay, which is the FDA-approved test for PD-L1, and it's shown here is an immunohistochemistry test. And we scored the proportion of tumor cells expressing PD-L1 versus the number of mutations. And as you can see here, we could not see a positive relationship between a mutational burden and PD-L1 expression, indicating that these metrics are independent from each other, and also supporting that we may be able to combine biomarkers to predict response better. Then, another important point here is that after being measuring T cells for many years, we realized that it may not be only about the number of T cells that we see in the tumors, and that's why we created this functional panel. And what I'm showing here is that we measured the number of T cells and the number of proliferation of T cells and granzyme. And interestingly, what we could see is that some patients or tumors have actually very few T cells, but they're very proliferative or active. In some cases have a lot of T cells, but they're not really doing much. So this is very important because the current standard to assess immune cells is just counting the amount of cells. But we don't really know if they're doing anything meaningful or they're just sitting there. So I think incorporating functional markers to this type of analysis is very relevant to understand the functional implications. So we applied this panel first to a retrospective cohorts of patients that did not get immunotherapy. So we wanted to know what happens here in patients treated with standard of care chemotherapy, which is usually platinum-based. So what we could see is that, as expected, patients with a lot of T cell activation and proliferation, which are indicated here with blue, did better than patients having low T cell activation or proliferation. But the other important point is that we could see that patients that had proliferation of T cells and activation of T cells were not the same, which is known in the immunology landscape. We know that T cells don't like to proliferate and produce granzyme at the same time. Usually we see one or the other. But this actually supported that a lot of activity of T cells in the tumor is associated with better outcome. So then we look at the patients treated with immunotherapy, and as you can see here, we see, again, very prominent difference between the presence of T cells and the immune or functional metrics, and then we correlated single markers with response to therapy. And interestingly, only the pre-existence of CD3 positive T cells was significantly associated with outcome, which was mentioned before in melanoma. But here we're showing that if the tumor is inflamed before seeing immunotherapy, it's more likely to respond to therapy, which is not surprising. However, the question we wanted to dissect out here is, can we stratify this inflamed tumor into functional groups? So what we did here, we stratified the cases into no or poorly inflamed, meaning low levels of CD3, and then the ones that were inflamed with high T cells, we stratify in low activation proliferation and high activation and proliferation to try to discern what is the group of patients that benefit. And what we could see here is that this is an example of an immune desert or cold tumor, very poor in T cells. You see that there is very little red here. This is an example of a tumor with high T cells, but low activation and proliferation. So this tumor is inflamed, but there is not a lot going on. And then there is an example of a tumor with high T cells and high granzyme production and T cell proliferation. So we had 3 different groups. But what we could see is that, surprisingly, the group that benefits more from immunotherapy is this group, what we call the dormant TIL phenotype, that it's a red curve here in the Kaplan-Meiers that shows actually a significantly better progression-free survival and a significantly better overall survival. And this is very interesting because the initial hypothesis was that the very active tumors will be the ones responding. But now it makes so much sense because in this case, there is not response expected. There are no TILs. In this case, the T cells are already fine, so the mechanism might be a different one. But in this case, we have T cells, meaning that they are able to recognize antigen, they're able to migrate to the tumor, but they are being silenced in the tumor microenvironment. And in this case is where we have more room for re-stimulation of T cells using immune checkpoint therapy. So I think we're narrowing down this population now to define what are the immune landscapes that are connected with immunotherapy. If this was true, and we could actually reinvigorate these dormant T cells, we will be able to demonstrate that experimentally. So what we did here next, we used a model called an immune PDX, and this was a collaboration with Miguel Sanmamed from Liping Chen's lab. So what we did here, we engrafted human tumors, complete tumors, including tumor and stroma, into immune-deficient animals, and we treated them with PD-L1 in the mouse so that we could measure the T cells before and after PD-1 blocker. And what you're seeing here is the result from that study in which we could see that upon stimulation with anti-PD-1 in a human tumor engrafted in an immune-suppressed animal, we could actually induce T cell proliferation and activation, and we went from this dormant T cell phenotype to an active T cell phenotype, and this happened only with anti-PD-1, but not with controlled PVS. So we were essentially demonstrating, and now mechanistically, that this is happening after PD-1 blockade. Then we got more ambitious and we used CyTOF analysis, which is essentially a mass spec-based flow cytometry to characterize these populations of cells. And as you can see here, the population that proliferates the most, it's typically positive for CD4, so they're CD4 helper T cells, and they express high levels of TBET, TIM3, and HLA-DR, which is a sign of T cell activation. However, the cells that produce more granzyme are predominantly CD8 effector cells, which is expected, but they actually express a lot more eomesodermin than TBET, indicating that they have a memory T cell phenotype, and they also express HLA-DR. And then we find a population that does both. They proliferate and they produce granzyme, which are not, typically they're positive for CD8, so the majority of them are effector cells, and they also have a different immune makeup. And now we're trying to dissect out these subpopulations more by using both functional and phenotypic markers. But we here demonstrated that there are different populations doing different functional aspects. The next important question was to try to understand if those immune phenotypes were actually correlated with the mutational burden. And actually, are we picking those tumors that are hypermutated using these functional T-cell assays? And as you can see here, the answer is no. So we actually plotted the number of mutations versus the type of immune cell phenotype, being the type 2 being the dormant phenotype. And we see that actually most of the hypermutated tumors are in the type 1, which are the non-inflamed tumors. And this was also unexpected. We expect the hypermutated tumors to be very inflamed. And this was also true for candidate neoantigens. What you're seeing here is a correlation between the amount of CD3 positive T cells and the number of non-synonymous mutations. And there was no positive relationship, actually a clear trend towards a negative relationship, which suggests that tumors that are hypermutated are very good at evading the immune response, and they don't really overlap. To support this further, we actually look at RNA sequencing data from the Cancer Genome Atlas. And what you're seeing here on the left are the lung adenocarcinomas from TCGA, and on the right, lung squamous carcinoma from TCGA. And here we plotted the mutational burden on the x-axis versus a number of immune cell markers, including T-cell markers like CD3, 4, and 8, functional markers, and even B-cell markers. And what you're seeing here, again, is a confirmation of a lack of relationship between the number of mutations and very important immune metrics. And this is particularly pronounced for lung squamous carcinomas, in which we essentially see a flat curve for everything. And we actually confirm here that the amount of CD3 transcript is not linearly related with the amount of mutations, supporting a lack of connection between these metrics. The next important question is what was the overlap between these TIL patterns and the PD-L1 expression. So we did, again, the data with the PD-L1 22C3 assay, and as you can see here, there was no significant difference between the levels of PD-L1 across the TIL groups. Again, highlighting that this is another independent metric. So now we're collecting a number of different pieces, some of them at the genomic level, some at the immune level, and some with PD-L1 that can potentially be integrated. So then, to prove that we could actually integrate these metrics, we use a bioinformatic model called PAR-DSA, which is a partition algorithm that does a regressive function. So essentially go from the outcome to pick up what are the variables that weigh more into that function. And as you can see here, looking at this, we input a number of clinical and molecular variables. And we could see that the algorithm suggested that the number of mutations, more than 354, and the number of smoking packs per year, more than 20, and then T-cell proliferation and the amount of T-cells were actually all very important to select for response. And we integrated all of these metrics. We could misclassify only 2 patients. So 47 or 49 were classified correctly to responder or non-responder. And when we look at the Kaplan-Meiers of this function, they're much better. Importantly, this is a small study and is overfitted because we trained the model in the same patients. But now we're validating these models prospectively to actually demonstrate that we can integrate both genomic metrics and immune metrics. So the next question, in the next few minutes, I will show you where are we going with this. So the sequencing, it's kind of figured out. We sequence well, we understand what we're doing, but how can we understand more about the immune composition? And one important question is, are other immune cells relevant to these responses? And how can other immune cells modulate T cells and how they interact with the tumor? So one important point that we have been trying to narrow down on is myeloid cells. And this is a study done by a fellow in my lab, Brian Hennig, and we came up with a panel to stratify the so-called good myeloid cells, or M1 type macrophages, from the ones that are immature or bad, which are usually called M2, and then also from myeloid-derived suppressor cells, as shown here, using functional markers. So we can stratify all the myeloid cells with CD11b, and then we look at CD68 for good or classically activated T cells, and then the absence of HLA-DR indicates a more immature phenotype or incapacity to present antigens. So we can now start figuring out immune cell populations. And we have actually validated assays to do this using multiplex fluorescence, and these are two lung adenocarcinomas. And you can see here that this one, for example, has a lot of CD68 positive macrophages that are indicated here with red. Tumor cells are indicated with white, as opposed to this one that has a lot of CD11 positive myeloid cells that are not CD68 positive, and they actually show different levels of HLA-DR. So now we are seeing tumors that have a different myeloid composition. And we are trying to understand what this means and how that could explain and correlate with the genomics of those tumors and the likelihood of response to therapy. Just as a proof that this is actually useful, we look at the retrospective cohorts of 250 patients, and we could see that just looking at CD11b, we can stratify patients by survival. And this is important because CD11b is expressed predominantly in immature myeloid cells, and it's downregulated in mature CD68 positive myeloid cells. So we're now understanding that patients with more immature populations tend to live shorter. The other important point is that the immune system runs on cytokines and chemokines. And those targets are very difficult to measure at the protein level because they're secreted very fast. So by the time we look at the tumor tissue, they might have already been secreted, and that secretion happens at different time points in different tumors. So really, we cannot, even if we can measure them reliably, we don't know if they're reflecting actually the state of the tumor. So to figure out this problem, we generated assays to measure interleukin or cytokine RNAs, because the RNA is upregulated, but it's not secreted. So by looking at the RNA, we can have estimation if those cytokines and chemokines were up or down regulated. And this is an example of a study that was recently published in which we look at the prediction value of measuring interleukin-A RNA, and we multiplex that with a positive control for RNA just to show that the paraffin tissue has measurable RNA, and then we colocalize with cytokeratin protein to know if the RNA is produced in the tumor or in cells that are outside the tumor area. And this is just the validation of the test in cell lines, and this is how it looks merged. And we could actually show that the majority of interleukin-8 produced in cancer patients treated with immunotherapy, it comes from the tumor, and it's actually predictive of, or early predictor of response to therapy. And this is published in this journal. The other important point is that we're trying to expand this effort and validate assays that work in paraffin tissue so we can look backwards and to measure additional transcripts. And this is just an example of another multiplexing assay in which we're measuring simultaneously DAPI for every cell, ubiquitin C as a positive control, then we measure PD-L1 RNA, interferon gamma RNA, and then we have the merge image. And these are cell lines without PD-L1 and transfected with PD-L1, and then we see PD-L1 showing up. And then these are PBMCs, or peripheral blood mononuclear cells, that we can stimulate in vitro using CD3, CD28. And then we can see upregulation of interferon gamma, which is not present in unstimulated cells. And this is all working in paraffin tissue. So we have validated assays to interrogate tissue, and this works in any collected sample. And then also, as expected, high levels of interferon gamma in a retrospective cohort are highly associated with better outcome, and these are the survival curves from another retrospective cohort, including 260 patients with lung cancer. And we could see that patients with high interferon, reflecting an active anti-tumor immune response, are actually surviving longer. Finally, in the last few minutes, I would like to show you where are we going with this. So we're very happy doing this 5 or 6-plex multiplexing, but we want to learn more. We want to know more about the immune cells, targets, and how we can understand the tumor microenvironment. So one of the important problems we have with fluorescence is that we can only measure up to 5 to 8 markers using spectral mixing because we have only a few fluorescence channels. The visual spectrum of fluorescence is only limited, and we can sort with filters, but we can't do much. So what we're doing now, we're moving from fluorescence to mass spectrometry. And this is called imaging mass cytometry, or IMC technology. And essentially what we do, we label antibodies with rare earth metals. And those metals are not normally in the human tissues, so we have very little noise. And by labeling those antibodies, then we incubate the paraffin tissue, and then we ablate each micron with a very sharp laser at 213 nanometer. So each ablation produces an ion cloud containing metals that are proportional to the amount of proteins. And then they're flowed through an inert gas chamber and then resolved by mass spec. And then they're reconstructed into an image. So now we can actually measure many more markers with spatial resolution, 1 micron resolution, and now make sense of what's going on in the immune tissue and the immune microenvironment. So this is the machine itself. This is my postdoc, and this is the machine. This is the microscope, and this is the site of Helios, which is a mass spec device. And this is a zoom in from the stage. And as you can see, this is a regular paraffin pathology tissue. And then we stain all together, and it's analyzed using this mass spec device. And this is how it looks. So it's very difficult to put many markers. Sorry, I think I skipped one. So this is an example of a human tonsil stained. These are 15 markers, and this is lung cancer stained with 15 markers, and this is before and after the laser ablation. The most important point is it's very difficult to look at more than 6 or 7 markers together because everything starts looking white. So we separate that into different panels. And as you can see here, as expected, most of the CD3 positive T cells are in the interfollicular areas of the tonsil, and most of the proliferating cells are in the germinal centers. And these are just like controls, and we did a lot of validation. And then we have tumors that are positive for cytokeratin, and we can see T cells in tumors and so on. So we are now understanding much better what's going on by looking at more functional markers. So we, over the last year, going back and forth and validating and invalidating reagents, we put together a panel that we call the immune oncology panel. And we included a number of structural components that we can use to do segmentation of tissue. So we have nuclear markers like histone 3, membrane markers like lipo-R, and then housekeeping genes that we can use to separate bioinformatically the tissues. Then we have a number of phenotype or immune markers, including T-cell markers like CD3, FORT8, FOXP3, a B-cell marker, CD20, a macrophage marker, CD68, a memory T-cell marker, CD45RO. Then we have a number of functional markers like granzyme proliferation markers, TBET and CD25, and beta-2 microglobulin. And then we have a number of targets. And this is very important because now we're looking at candidate targets to predict response to therapy. And here we included PD-L1, PD-L2, a number of other B7 family members, IDO, PD-1, TIM3, LAG3, and so on. So we're spanning a number of different things, and this is all measured together. And to prove the value of this panel, we actually did a pilot study in which we are trying to demonstrate what is the difference between the normal lung and the tumor from the same patient. So what we did here, we collected tumor, as shown here, and then non-tumor tissue that was at least 5 centimeters apart. And this is the way they look on the HD. So this is normal lung or non-tumor lung, and this is lung adenocarcinomas. And we look at the difference using this imaging mass cytometry panel. And what you're seeing here is our representative captions from a normal lung, which is not surprisingly less cellular and with a lot of airspace, and a tumor. So we actually realized that we had to normalize by the amount of cells. So we started normalizing by the number of histone 3, which is a nuclear marker. So we can correct for the amount of cells in each preparation and make this assay comparable. And then this is a study in which we are showing all the normal lungs on the left heat map side and then all the tumors. And these are each of the structural, phenotype, functional, and regulatory markers. And what you're seeing here that we can see already heterogeneity across the normal lungs, but a lot of variation across the non-small lung cancer cases, and a number of things that are upregulated in lung cancer as opposed to the normal counterpart. And we start seeing, for example, a number of immune cells like CD8, we see a number of checkpoint markers like PD-L1, PD-L2, and so on. So now we can not only map the targets, the immune composition, but we can compare across different tumors and across different normals. And then the most important point, and this is a quantitative method, so we can measure the amount of protein in these tissues and make statistical comparisons. And this is just the stratification of this panel, and you can see that we saw significant upregulation of CD8 positive cytotoxic cells, positive for CD45RO in lung cancer as compared to normals, but also functional markers like TBET, which is a transcription factor of the STAT pathway, and then beta-2 microglobulin, and then also a number of targets expected to be in the tumor like PD-L2 and IDO1, and then a number of other markers like PD-1, LAG3, and TIM3, and VISTA. So now we can really understand much better, and not only in the tumor, but looking at the normal allows us to look at the candidate therapeutic window, because we don't want to target things that are upregulated also in normal lung, because that could induce toxicity. So we can start exploring the benefit or relative benefit of using blockades. Then we can compare pairs in normal tumors. So what we did here, we wanted to look if the associations were consistent. And we could see that by and large, the relationship for the immune markers were very consistent. A few of them, like granzyme, were a little bit up and down. But in general, we see consistent association between the normal tumor pairs. And then since this technology has this spatial resolution, we can actually do single-cell analysis. So what we did here, based on the expression of CD3, we randomly collected individual cells that were positive for CD3. And this is the plot of CD3 positive T cells in normal lung versus lung cancer. And this is interesting because we could see the heterogeneity of T cells in normal lung and the variation in lung cancer. But when we compare the markers only now in T cells in both tissue types, we can see that T cells in lung cancer express higher levels of CD45RO as seen before, but also candidate markers like PD-1, LAG3, and things that we can now actually action their clinical trials against this therapy. So we can understand much better the likelihood of those tumors to benefit or can potentially benefit from specific therapies. We can do the same for tumor cells now. So we selected cells that were positive for cytokeratin, and the same thing, we compared normal type 2 pneumocytes from normal lung versus cytokeratin positive tumor epithelial cells, and we could see significant upregulation of proliferation as expected, but also of candidate targets like PD-L1 and CD47. So we, again, see patterns of immunomodulatory therapies both in the tumor cells and also in the immune side. So I think in conclusion, after all of this, I hope I convince you that biomarkers can be used to support the optimal clinical use and development of immunotherapy. In other words, we should be using this type of studies to drive the next trials and not do every trial and then try to make sense. I think I've tried to convince you that using quantitative assessment that are objective is better than just do estimations or qualitative assessments. And the second important point is that genomic and immune phenotype things can be integrated to understand sensitivity and resistance. So this is not only about the tumor and EGFR mutations, it's about how the immune composition is recognized by the immune system, and then what does the tumor to overcome that immune pressure. And from that, we learned that in lung cancer, mutations and candidate neoantigens are independent from the tumor immune infiltration and PD-L1 expression, and the elevated number of T cells, but with low signs of activation and proliferation, what we call a dormant T cell phenotype, are the most likely to benefit from PD-1 axis blockers. And finally, I would like to close saying that additional strategies to interrogate the tumor immune microenvironment are emerging. I show you some examples of RNA immunoprofiling, multiplex, high-order multiplexing, and there's a lot of efforts in circulating biomarkers, which I didn't talk about, but I think the future will go in that direction. And I think we should keep the eyes open to not only look at the tumor, but look at the immune system to understand and enhance these therapies even more. With this, I would like to thank all the people actually doing the job, the people in my lab. The ones that are in black or in bold are the ones that did most of the results I presented. People from the Yale immunosequencing group, particularly Jungmin, who is the bioinformatician, who is dealing with all of this HLA typing, neoantigens, and so on. And then collaborators at Yale, and then obviously the cores and funding sources. And finally, I would like to say that I will be happy to discuss further. I'm not difficult to find, so that's why I included this. If you Google my name, this will show up. There is a link to my lab, and I'm happy to answer questions and keep connected. And thank you.